All right, so this is the video for the STEM station. You should have already read your article, taken some notes on your note sheet. You need these two cut out, and then you also need your model of the vascular system that you have already made. Okay, I cut mine out. You do need to cut out these little insides, so I was just going to very quickly show you how an easy way to do that. You bend it, you don't fold it in half. Cut a hole in the middle, and then you can just kind of cut very quickly. So if we cut, and then once you have both of these cut out, you can continue with the next step. Okay, so now that you have cut out the middles, we're going to use them like little windows into the interior of the plant. As you can see, these are both made out of cells because we are talking about tissues here. In this case, we're talking about dermal tissue, which is the skin. So on your body, this is dermal tissue. You have 3 layers, the epidermis and then the 2 bottom ones. Plants also have an epidermal layer, and then they have an inner dermal layer, that subdermal. It's just consisting of different types of cells. Okay, that's why one is light green and one is dark green. So we are going to take our model, and you're going to need some tape, and this is super easy. You're going to start with the light colored one because the light colored one has the smallest window. So you're just going to line it up at the bottom, somewhere where you can see both the xylem and the phloem through the window. And then you're going to just wrap it around and tape it. Wrap it around, tape, wrap it around, and tape. You will have some straw sticking up at the top. That's okay. You can cut it off later if you want to. All right, so now I have a subdermal layer of cells, tissue. I can see my xylem and my phloem that are inside this little casing. And now I'm going to add my epidermal layer, the outside of the skin. So this is, anytime you look at a plant, this is what you see. Bark on a tree, the green part of romaine lettuce, any of those things. So you're going to take this one and you want to layer it so that you can actually do it either way, really. I'm going to put mine so you can see the green layer at both the top and the bottom, and then you can see the xylem and phloem on the inner side. So again, you're just going to wrap it around. There you go. Take some tape. Tape that together. Take this tape. Tape it together, and now you have a completed stem. You have the inner layer of your stem, which you can see there's xylem and phloem. Then you have your ground cells, your xylem and phloem, ground cells. You have your inner layer of dermis, and then you have your outer layer of epidermis, and they're one wrapped around the other. So when you look at it from the top, you look down into it, you can see the xylem and phloem, the ground cells in the middle, then you have an inner layer of tissue, and then an outer layer of tissue. Okay? Once you have completed taping this together, you're going to need to add some notations from your notes. So anything that is from the things that you wrote down from the questions to consider, you can add little flags to it or tape it on a piece of paper and tape it next to it. Information to label your model.